So we left off last time talking about the anatomy of the kidneys, and I gave you the basic definitions of all of these physiological processes in the kidneys. Today we're going to go over them in nitty-gritty detail, but let's just review. Glomerular filtration is the passive transport process that occurs in the renal corpuscle, where the hydrostatic pressure in there is increased due to the difference in size between the afferent and efferent arteriole. So we put a lot of pressure on that blood in the glomerulus and push against those fenestrated capillaries, which are super permeable. So we push everything out except large proteins and cells, and we get all of this filtrate collected that is in chemical equilibrium with blood plasma. So that filtrate then is going to enter the renal tubule, and that's where we have our proximal convoluted tubule, our nephron loop, and our distal convoluted tubule. And throughout the renal tubule, we have both tubular reabsorption, which is going to reclaim things from that filtrate that was produced here. So we'll reclaim things that we need and bring them back to the blood. And then we'll do tubular secretion also throughout the renal tubule. And what's happening in tubular secretion is we're pulling everything out of the blood that we don't want and concentrating it so that we can excrete it in our urine. So there's going to be passive and active transport processes going along throughout the entire system. I'm going to go over the general movement of our ions and materials through each part. So glomerular filtration, the first process in urine formation. This is actually the most important for determining the rate and kind of concentration or amount of urine that you're going to produce. So this determines the rate of urine, or glomerular filtration rate determines the amount, the rate of urine formation, or the amount of urine we're going to produce. And glomerular filtration is occurring across a structure we call the filtration membrane. And this is right there again in the renal corpuscle. So the filtration membrane has three parts. It's got the fenestrated glomerulus, so it's a simple squamous epithelial tissue with our pores, those fenestrations, and then we have our visceral layer of the glomerular capsule wrapping around there, and it's those podocytes wrapping around creating that. And then there's a basement membrane shared between them. So what we could say the parts of the filtration membrane are the endothelium, or the glomerular capillary, the basement membrane, and then the visceral layer of the glomerular capsule. And again, that is those podocytes wrapping their feet around to kind of give some structure to that glomerulus, so that we're not just going to be pushing out cells and proteins every time we have a change in blood pressure. So, okay, that's the filtration membrane, and this is the site across which glomerular filtration is going to occur. And what happens as these podocytes wrap their feet around these glomerular capillaries, we get the creation of these things called filtration slits. So, filtration slits, and that's going to allow for some control of movement of materials out of the blood. What do I mean by that? Well, I can, by wrapping around and creating this like filtration slit, I can allow for smaller things to get through, but I can like control and allow larger things to stay in the glomerulus. So if I were to imagine what's going on right here, this would be the other side of my glomerulus, and I have blood cells coming through here, and there are large proteins and things. Those are all going to get stuck and stay in the glomerulus, and they'll exit in the blood in the efferent arteriole. All of the rest of our materials are going to be pushed out now and collect in this fluid that's going to become filtrate, and this filtrate is going to move through the renal tubule and be adjusted to become urine ultimately. So when we leave this glomerular filtration process, we've reached chemical equilibrium between these two things, but this blood still has large proteins and its cells in it because this filtration slit is allowing for some control of that, creating some stability and structure there so we can keep those things in there. We'll talk more about what you can find in urine and what it can tell you about the health of the system and everything in a little bit. To understand how filtration is working, we need to understand the pressures in the system that are causing the movement of our fluid. So if we look, I'm going to draw my glomerular capsule here. This is my capsular space, and this is going to collect my forming filtrate. And I kind of get nitty-gritty about the usage of different words for the fluid that's moving through the system, because we've got two fluids that we're actually tracing. We're tracing the flow of blood into the kidneys and then into out the glomerulus and out. But we're also then tracing the movement of urine. So we've got to follow blood and we've got to follow urine. And if we look here, this filtrate that is going to be the first fluid that we form is at chemical equilibrium with blood plasma. So out here in the blood plasma, we still have things we don't want, like wastes and drugs and excess hormones or whatever. So I need to be able to pull that out and concentrate it. We're not doing that here. That's going to happen when we move through the renal tubule. I'm establishing the beginning of the movement of these fluids, and when I'm here, my filtrate is at chemical equilibrium with blood plasma. As soon as I get to my proximal convoluted tubule, I'm going to begin tubular reabsorption and tubular secretion. So I'm not really filtrate anymore, although a lot of books still tend to call it filtrate. I'm not urine yet. I'm not urine until I get down to those little papillary ducts at the end of this whole system. And I can't reclaim any more water. So I will probably call this fluid moving through here tubular fluid, as it's moving through the renal tubule. And right now, the only time that I really call it filtrate is right here when it's collecting in this capsular space in the renal corpuscle. So just be aware of that. What do we got going on? We've got blood plasma, and if we think about what the plasma coming in here has, it's got everything. It's got nutrients, wastes, drugs, ions, all of that. A lot of water, so it contains water, nutrients, any drugs we might have in the system, vitamins, minerals, what are all those, ions, all that stuff. Everything that's in our blood is in blood plasma. So this first fluid that's produced, filtrate here, is at chemical equilibrium with that. The only thing that's missing from filtrate is the big cells and big proteins that get stuck here because they can't get past that filtration slit. So my filtrate, we'll say, is at chemical equilibrium with plasma. And then my tubular fluid, I'm going to immediately begin pulling stuff out of it that I want to bring back to the body and concentrating stuff in it that I want to take out of the body. So that's going to be different. But right now we're talking about glomerular filtration and we're looking in here. So what are our pressures in this system? So we've got inward and outward pressure. So the outward pressure is going to be the pressure of the blood on that glomerular capillary. It's hydrostatic pressure because it's a fluid, and it's the hydrostatic pressure of the glomerular capillary. So my outward pressure is hydrostatic pressure of blood. And if we think about it, there's hydrostatic pressure of blood in this arterial coming in here. and it's getting dispersed, but this is like the last pressure due to ventricular systole. And when we come in here, since this afferent arteriole is bigger than this efferent arteriole, the hydrostatic pressure in the glomerular capillary is actually higher than this, so that we get a lot of pressure. I love this about the nephron. Like, it's just perfectly like organized to lead to its function. We get a lot of pressure in there. We can push all this stuff out so that in this filtrate now, I can start moving this through the rest of the system and make adjustments. So that outward pressure out is high. This is the hydrostatic pressure of blood in the glomerular capillary. Let me abbreviate this HPGC. And it's the hydrostatic pressure of the glomerular capillary, and it's somewhere around, normally around 55 millimeters of mercury out. Okay, hydrostatic pressure is just the pressure of a fluid on a membrane. Filtrate is a fluid, and it's going to push back onto that glomerular capillary. But this is the hydrostatic pressure now in this capsular space. So that's going to be a little bit of an inward pressure. So we've got an inward pressure that's also a hydrostatic pressure. But this is the hydrostatic pressure of the filtrate on the glomerulus. And we could call this our HP, hydrostatic pressure of the capsular space. And I feel like that's around somewhere about like 15 millimeters of mercury in. So it's about 15 millimeters of mercury back in. Okay. And then I also have another inward pressure. It's osmotic pressure that's created here in the glomerular capillary. Why is there osmotic pressure here? Well, because this blood seems hypertonic. If what I did here was create this system where I was at chemical equilibrium between these two fluids, with the exception of blood having these large cells and large proteins, then now blood feels hypertonic. So water wants to come back in. So this is actually a pretty high inward pressure at about 30 millimeters of mercury. So the other inward pressure is osmotic pressure in. And we could say this is due to the blood seeming hypertonic, or I'm actually going to say it's due to the hypertonicity of the blood. And we could put in parentheses because cells and large proteins normally don't get filtered out. So I'll say shouldn't get filtered out. Okay, so, and again, it's around 30 millimeters mercury in. So then we have, if we take all of this into consideration, we get what's called our net filtration rate. NFR is our net filtration rate. And this is going to set, like you could think of it as our glomerular filtration rate, GFR, and this is most important for like setting the rate of urine formation, which I already wrote up there, so I'm not gonna write it again. Okay, so our net filtration rate, how can we calculate this? Well, we take our outward pressure and subtract the inward pressure and figure out what are we going to get. So what did we have? We said we had an outward pressure of around negative 50 or around millimeter. We said we had an outward pressure of around 55 millimeters of mercury out. And then we've got around 30 millimeters mercury in due to osmotic pressure. and we have around 15 millimeters of mercury in due to hydrostatic pressure of the capsular space. So our net filtration rate is around 10 millimeters mercury out. And we want to keep that pretty constant. We want to keep it set at around 10 millimeters of mercury out as our outward pressure, so that we have a set rate of urine formation so that we can keep our blood volume constant. Now, of course, if you're over or underhydrated, things are going to change, and we'll talk more about that in a little bit. But just when you're sitting around trying to be in homeostasis, then we want to keep our pressure, our net filtration rate at about 10 millimeters of mercury out so that urine formation is constant. It should hopefully all come together by the end of this lecture. Okay. So that's our net filtration pressure. And it's going to lead, I think I called it our net filtration rate. Net filtration pressure is going to lead to our glomerular filtration rate. And again, this GFR has to be kept pretty constant so that we can keep blood volume constant and urine volume constant so that we can stay in homeostasis. It is so important to keep glomerular filtration rate constant that the kidney actually has two mechanisms on its own that are going to control glomerular filtration rate despite changes in systemic blood pressure. So that if there's an increase in systemic blood pressure, we would expect then to see more pressure in here, and so we'd have more urine formation. That would drop our blood volume. We don't want to do that in response to just a small change in blood pressure. So the kidneys can, right there themselves, drop glomerular filtration rate so that we don't get too much urine production, and vice versa. Then there's also a mechanism by which we can, those macula densa cells we were talking about last time, can also sense what's going on with glomerular filtration rate and help to adjust it. So this is super, super important. We're going to look at the intrinsic controls that are going to help just to maintain urine production and urine volume. And then we're going to look at what we can do to glomerular filtration rate extrinsically to help to maintain systemic blood pressure. So intrinsic controls are going to keep glomerular filtration rate constant for urine production, and extrinsic controls are going to adjust glomerular filtration rate to maintain blood pressure. Okay, let's talk about all of that. Teaching from my living room is challenging, so I'm never sure if I'm making all the sense that I feel like I would in a normal class. Although maybe in a normal class I don't make all the sense that I think that I do. So let's just be clear and be sure that we're on the same page about everything that I just talked about. So, because we're going to get in here now and look at setting glomerular filtration rate. I'm not horribly concerned that you calculate net filtration pressure, but I want you to understand these pressures so that you can understand what's happening in the system when we have to kick in our various intrinsic and extrinsic controls. So again, we said our pressure outward was about 55 millimeters of mercury out. And this was our hydrostatic pressure of the glomerular capillaries. We said we had two inward pressures, the osmotic pressure of about 30 millimeters mercury in, and this was due to our osmotic pressure of the glomerular capillary due to the hypertonicity of blood. And then we had one more inward pressure, which was the hydrostatic pressure of the capsular space that had our filtrate developing in it, and that was about 15 millimeters of mercury in, which left us with a net filtration pressure of about 10 millimeters mercury out. And we want to keep this constant to maintain homeostasis so that we aren't just all of a sudden rapidly changing urine volume and thus blood volume in response to changes in small changes in blood pressure and vice versa. So what's going on here? Okay, our intrinsic controls. There are two ways that if there's a change in glomerular filtration rate, which is going to be determined by this pressure. So again, glomerular filtration rate is the most important for setting the rate of urine formation, and thus like the volume of urine that we're excreting, and thus the volume of blood that we have. So we want to keep this constant, or relatively constant. We want to stay in a pretty tight homeostatic range. So the intrinsic controls are the ways that the kidney has just here, right here, at this juxtaglomerular apparatus that we talked about last time, so that it can adjust glomerular filtration rate and keep urine production constant despite small changes in systemic blood pressure. So there are two ways that it can do it. There's what's called the myogenic feedback mechanism and one that's called the tubuloglomerular feedback mechanism. So let's talk about both of those. Our intrinsic controls, I'm actually just going to write it up here. So for intrinsic controls, of glomerular filtration rate, we've got two. Let's just talk about them one at a time. The first is called the myogenic mechanism. And if we look at this word, myo, muscle, genic, generate, this is generated by the smooth muscle in the afferent arteriole. So if you recall last class, we said there was this thing, the juxtaglomerular apparatus, and that involved these macula densa cells that were wrapping around and touching this afferent arteriole. So we'll talk about that as this tubuloglomerular feedback mechanism. But when we were talking about that, we said these cells in the afferent arteriole, the granular cells, are acting as baroreceptors, pressure receptors. They're detecting changes in stretch. And so if there's a change in stretch here, they're going to be able to either increase or decrease the diameter of this afferent arteriole to keep GFR constant despite what's going on in the system. What the heck am I talking about? Okay, well, let's just imagine. Let's imagine for a moment that you have an increase in blood pressure. And so a slight increase in systemic blood pressure. So we get an increase in blood pressure. What is that going to do to the stretch here in the afferent arteriole? Increase it or decrease it? It's going to increase it. Okay, let's imagine this then. So now let's just not change anything yet. Let's walk all the way through what would happen, and then we'll see why we need to fix it. So let's imagine that out here before I had about 10 millimeters of mercury pressure out. Well, what if I have an increase in blood pressure that's so great that it increases my hydrostatic pressure in the glomerular capsule, from the glomerular capillary out, so that now instead of being 55 millimeters of mercury out, I'm 60. So now I'm 60 millimeters mercury out. Well, my osmotic pressure in isn't going to change. My hydrostatic pressure of the capsular space in isn't really going to change. So now what is going to change is I've got about 15 millimeters of mercury as my net filtration pressure. So now I'm going to make more urine, and it'll happen faster. And that's what would happen if I don't fix it. And it was just because I had this increase in blood pressure, and now I'm going to make more urine. So sure, over the long haul, if you've got increase in blood pressure, that's what will happen. But if this is just a quick change in systemic blood pressure, I don't want to change my urine production and my blood volume. So what I can do is, by the myogenic mechanism, I can constrict this smooth muscle, myogenic, and now I can decrease it so that my hydrostatic pressure out stays where it's supposed to. And it was just because in response to this change in stretch, I constricted my smooth muscle. So let's write that out. For my myogenic mechanism, what would happen is we would get an increase in blood pressure. This is going to increase stretch in the afferent arteriole. This is going to be detected by my JG cells. They are going to respond and stimulate constriction of the afferent arteriole. And this will maintain glomerular filtration rate despite this small increase in blood pressure. Let's think about it the other way. So now we're sitting here at rest, and we have about 55 millimeters mercury pressure out due to hydrostatic pressure in the glomerular capillary, and then my blood pressure drops. What's that going to do to stretch? It's going to decrease the stretch, right? So it'll decrease the diameter of my afferent arteriole. So what happens then? Okay, so I get a drop in blood pressure. This is going to decrease my stretch of my afferent arteriole. So what is that going to do to my hydrostatic pressure in the glomerular capillary? Increase it or decrease it? It's going to decrease it, right? Because now I have a smaller difference between my afferent and efferent arteriole. So if I have a small drop in blood pressure, then I decrease this difference, and I decrease my hydrostatic pressure in the glomerular capillary. So let's say, I don't know, I come down to 50 millimeters of mercury. And then I come down to only 5 millimeters of mercury as my net filtration pressure. So then I don't produce as much urine, and now I have more blood volume. But if this was just a small change in blood pressure, I don't want to do that. Okay, so let's finish writing it out. A drop in blood pressure is going to decrease the stretch of the afferent arteriole. What this does is it decreases the hydrostatic pressure in the glomerular capsule, or in the glomerular capillary, which is going to drop our net filtration pressure, which would drop our GFR. If we drop our GFR, we drop our urine production. We don't want to do that. So this is sensed by those JG cells. And what do you think they're going to do to the afferent arteriole? Constrict it or dilate it? They'll stimulate vasodilation. So now we had this decrease in stretch. We'll stimulate vasodilation. I can increase in stretch, and I can keep glomerular filtration rate constant despite that small change. And I'm all good. So my decrease in blood pressure is sensed by these JG cells. They're going to stimulate dilation of the afferent arteriole, and GFR will remain constant, or we'll say remain in homeostasis. So my next intrinsic control, so something that is intrinsic to the kidney, is what's called the tubuloglomerular feedback mechanism. Tubuloglomerular feedback mechanism. And we talked about this the other day when we were talking about the anatomy, and I said that what happened in the body is that the distal convoluted tubule is going to flip back around and touch up against the afferent arteriole. And so the cells that are part of that and that are involved in this are those macula densa cells that are acting as chemoreceptors, sensing sodium chloride levels of this tubular fluid coming through here. So if glomerular filtration was occurring too quickly, or if glomerular filtration rate was too high, do you think you would have too much or too little sodium chloride in the tubular fluid passing through? You would have too much. And this is kind of weird. This pathway can get hard for people to wrap their head around. So I'll make sure that we write it all out. And if you have questions, you can drop them in the discussion board. But what happens is that we actually need to reabsorb a bunch of sodium chloride, because sodium is going to have the biggest impact on osmotic pressure, and we need a lot of water. So we're going to reabsorb sodium. Water is going to follow it. It's important. So if we've got a lot of sodium coming through here in this distal convoluted tubule region, then we know that, okay, the glomerular filtration is occurring too quickly. We're making urine too fast. I need more time to reabsorb, so I can slow glomerular filtration rate so that I've got more time to reabsorb my sodium chloride. And then that'll drop in the tubular fluid moving through, and I'll come back to homeostasis. Okay, so if I need to drop glomerular filtration rate, what can I do intrinsically? Can I increase or decrease the diameter of the afferent arteriole to drop glomerular filtration rate? I'm going to decrease it, right? So that I can decrease the hydrostatic pressure in the glomerular capillary, decrease that pressure out, decrease glomerular filtration rate, and keep my urine production, or slow it down so that I can have enough time to reclaim all of my sodium chloride. So I hope that was clear. Clear as mud. Well, let's see what's going on. Let's write it down. So what's going on here is that, let's say we have an increase in sodium chloride in our tubular fluid, which some other books are calling filtrate. I'm going to call it tubular fluid. So in here I've got an increase in sodium chloride. This is going to be detected by those macula densa cells. So they're the sensor. This is detected by my macula densa. And they're going to tell these extraglomerular mesangial cells, hey, glomerular filtration rate is too high. Like, there's not enough time to reabsorb this sodium chloride. We got to slow it down. Tell the afferent arteriole. So then the extraglomerular mesangial cells are going to tell my JG cells, hey, glomerular filtration rate is too high. Constrict the afferent arteriole so that I can drop my hydrostatic pressure out. So that will drop my net filtration pressure so I can drop my GFR and have some more time to reclaim that sodium chloride that's going through. So we're going to tell the JG cells to vasoconstrict the afferent arteriole. The effect of this is going to be then that we will drop our hydrostatic pressure in the glomerular capillary, which will drop our net filtration pressure, which will drop our glomerular filtration rate, and we'll have time to reabsorb sodium chloride, and then we'll come back to homeostasis. Well, what about if there's not enough sodium chloride? So what if that's, instead of sensing an increase in sodium chloride coming through here, we sense a decrease in sodium chloride. So if there's a drop in sodium chloride, then this is still going to be detected by my macula densa cells. And now if there's a drop in sodium chloride, what does this mean? Well, that my GFR is too low. I'm not making enough. So I am going to tell my mesangial cells, who are going to tell my JG cells, who are going to stimulate vasodilation in my afferent arteriole. So now I can increase my hydrostatic pressure out. I'll increase my net filtration pressure. I'll increase my glomerular filtration rate. And now I can get more filtration going through here so I can reabsorb more sodium chloride. And I come back to homeostasis. All right, make sure you try and work that out in your mind because I know these feedback loops can get tricky. So extrinsic controls of glomerular filtration rate are going to be the way that we can control glomerular filtration rate so that we can control urine production, so that we can control blood volume and maintain systemic blood pressure. So now this is going to involve the entire body, and maintaining systemic blood pressure is so important. Sometimes you have to override whatever the auto-regulation mechanisms want to do and just like to the detriment of the kidneys so that we can maintain blood flow to our more vital organs. So extrinsic controls are going to kick in and alter glomerular filtration rate so that we can maintain systemic blood pressure. So I'll kind of write that as a definition down here. Extrinsic controls help to maintain systemic blood pressure. And we can do that by altering urine output, which will have an effect on our blood volume. So our first extrinsic control will be like in response to a fight or flight situation where you're and you have like an extreme decrease in blood pressure. So let's say blood volume. You have a decrease in blood volume, which is going to decrease your blood pressure tremendously. I think it's called hypovolemic shock. Then you're going to be in this extreme sympathetic nervous system situation, and it's going to cause extreme constriction of the afferent arteriole. And if we cause extreme constriction of the afferent arteriole, what's going to happen to hydrostatic pressure in the glomerular capillary? It's going to drop, right? So then what's going to happen to my net filtration pressure? It's going to drop. What's going to help in the glomerular filtration rate? It's going to drop, and I'm not going to make urine. I'm going to keep all of that fluid so that I can maintain my blood volume. So for extrinsic controls, we could think about what happens when the sympathetic nervous system is activated. It's going to stimulate extreme vasoconstriction, both through norepinephrine as the neurotransmitter and through epinephrine as the hormone. So we'll stimulate vasoconstriction. And this is in the whole body, so that what we can do is we can increase total peripheral resistance, which is going to help to increase blood pressure. So if we're stimulating vasoconstriction in the entire body, the afferent arteriole is no exception. So by stimulating this vasoconstriction, we will also vasoconstrict the afferent arteriole. What this is going to do is decrease the hydrostatic pressure of the glomerular capillary, which was our hydrostatic pressure out. This will decrease our net filtration pressure, which will decrease our glomerular filtration rate, which will decrease our urine production, which will help us to maintain our blood volume. If we have more blood volume going through a system that has constricted vessels, then we're going to increase blood pressure. So in situations like a fight or flight situation where you need an increase in blood pressure, that's what you'll see. This is what will happen. So if we were thinking about a stimulus for this pathway, what would get this going? We could think, well, this could happen in response to a drop in blood pressure, like a systemic blood pressure that's going to be maintained and severely detrimental to the system if we don't recover. The other thing that we can do is the renin-angiotensin mechanism, and we've already talked about this a lot, so this should hopefully just be a review. So here, in response to a drop in blood pressure, now this should all be coming together. What's going to happen to the diameter of the afferent arteriole in response to a drop in blood pressure? It's going to drop. So this is a decrease in stretch of my afferent arteriole. This is detected by my juxtaglomerular cells or my granular cells, my JG cells, and they're going to release renin. Renin's going to combine with renin substrate coming from the liver. That's the angiotensinogen. That's going to give us angiotensin 1. That's going to go back to my heart, get pumped to the lungs where the angiotensin converting enzyme is and get converted to angiotensin 2. Angiotensin 2 is a vasoconstrictor. So angiotensin 2 is going to stimulate vasoconstriction. That will increase total peripheral resistance, which will help to increase blood pressure. But if I stimulate vasoconstriction here, then I decrease my hydrostatic pressure out here, I decrease my net filtration pressure, I decrease my glomerular filtration rate, I decrease my urine production, I increase my blood volume. More blood through thinner vessels helps me increase my blood pressure. So the other way that we can get this going is that if we've got this decrease in blood pressure that's going to do that, it would drop the amount of sodium chloride in here. So the macula densa cells can also sense that drop in sodium chloride in the fluid and stimulate the granular cells to release renin. So that's pretty cool. What else? I think that's all I want to say about glomerular filtration rate. So when we leave here now, we're going to begin active and passive transport processes that are going to establish the concentration of all of the various substances that we find in blood plasma and in urine. So what we would see is that when we exit this efferent arteriole, we're going to enter those peritubular and vasa recta capillaries. So now what we have in here is our blood plasma, and it still has a bunch of stuff in it that we don't want. It has all the excess drugs, it has all the excess vitamins, it has waste, everything that we're pulling out. And what we have in here was this filtrate that has a bunch of stuff in it that we still do want. It's becoming tubular fluid, so it's got glucose, it's got, I mean, if I'm low on vitamins, then I want to bring those back from here. So what I'm going to do is I'm going to start, as I move through the tubule system, I'm going to start pulling things out of the blood and concentrating into the tubular fluid. So if you look here, this arrow from the blood to the tubular fluid, that's tubular secretion. And then I'm going to take things from this tubular fluid and bring it back to the blood because I want it. That's tubular reabsorption. So that's where we're going. Tubular reabsorption is the process by which we bring back all of our wanted and necessary materials that got pushed out in filtration. So tubular reabsorption brings back needed materials that were pushed out in filtration. And we can do this through the cells, across the cells, through the transcellular route, or we can do this between the cells through the paracellular route. So now we have moved away from the renal corpuscle and have entered the renal tubule, which again has our proximal convoluted tubule, our nephron loop, and the distal convoluted tubule. So here in the proximal convoluted tubule is where we're going to do most of the reabsorption of our filtrate. So that reabsorption is happening if we look in here and zoom in here. Here's our tubular fluid passing through here. So which surface of these epithelial cells is this? The apical or the basal surface? This is the apical surface, right? And the basal surface is out here. So on the apical surface, through the transcellular route, I'll bring materials through my apical surface and pass them through here and then let them out on the basal surface, and those materials can then be picked up in my blood vessels. So my transcellular route uses vesicular transport, or things are transported from the apical surface of the cell through the cell to the basal side and released into the blood or into the interstitial fluid, and then can be then picked up in the blood. In the paracellular route, this is how materials are going to move between these cells that through their kind of leaky intercellular clefts. So this is how materials move between cells And different substances are going to use different processes you can read about specifically in your book. All right, so the active transport of sodium is going to set up the gradient that's going to allow for secondary active transport of most of our substances to be reabsorbed. So the movement of sodium is hugely important, and each part of the system kind of does it a little bit differently, so we're not going to worry about the nitty-gritty details of that, but we'll just say that that sodium is actively reabsorbed throughout the renal tubule, and it's going to have the biggest kind of pressure on the movement of water, help create this osmotic pressure gradient, but the active transport of sodium is going to set up the concentration gradients that allow for secondary active transport of almost everything else to be reabsorbed. So we could say sodium is actively reabsorbed throughout the renal tubule, and the primary active transport that is used to move sodium is going to set up secondary active transport for almost everything else. So we'll say it sets up the osmotic pressure gradient, or it really helps to set that up. So water's going to follow sodium, so it's got the biggest impact on osmolarity. So we'll say it sets up secondary active transport for almost everything else. So we'll say for many other solutes. What else do we want to say about it? That water is going to follow sodium. Okay. So what about water reabsorption? Where do we do water reabsorption? Again, throughout the renal tubule, depending on where we're looking, is going to determine how we're reabsorbing our water. So for water reabsorption, we could also say this is occurring in different ways throughout the renal tubule. And water reabsorption is either going to be obligatory water reabsorption, like water is obligated to go when it follows sodium, for example, or facultative water reabsorption that happens when we facilitate the reabsorption of water by doing something like putting antidiuretic hormone in the system and increasing the number of these things called aquaporins, which are water transporters. So for water reabsorption, we'll say again, this is happening throughout the renal tubule, and it's either obligatory, where our water is moving due to osmotic pressure, like when it follows sodium, or facultative, or facilitated. Different books use different words. So facultative. And what happens in obligatory is water is obligated to move due to osmotic pressure. So it's obligated to go, it's obligated to follow sodium. Sodium hypertonicity makes things hypertonic. And then our facultative reabsorption is going to be when we facilitate the movement of water by doing something like increasing aquaporins by adding antidiuretic hormone. So facultative is when water reabsorption is facilitated by antidiuretic hormone. And what this is going to do is it's going to increase these water transporters called aquaporins. Okay, and then other solutes are going to be reabsorbed according to the body's need. And again, this is going to primarily be driven through secondary active transport that is established. The concentration gradients are established during the primary active transport of sodium. So for other solutes, we'll say they are reabsorbed as needed. And Most often, secondary active transport is used. What are other things that we're going to reabsorb? Glucose, amino acids, vitamins, minerals, all of those things that are nutrients. Those are going to be things that we're going to want to bring back into the body. And so those will be brought back in, again, according to need. So let's talk in greater detail about where each of these things is occurring and what we're reabsorbing where. And then we'll talk about secretion next. Okay, so the proximal convoluted tubule is proximal to the glomerulus from the fluid perspective. So most of the adjustments to this tubular fluid that's going to become urine are being made here in the proximal convoluted tubule. If we look at the cells in the proximal convoluted tubule, they're these columnar cells that have a lot of microvilli. The apical surface is up here facing the tubular fluid. And so we can put a lot of transport proteins in there so that we can get that active transport of materials. So it's going to reabsorb like 65% of our filtrate. And so we'll be taking this material out of the tubular fluid passing through and bringing it back into the blood. So what are the things that we are going to want to reclaim? We're going to reclaim 100% of our glucose. We're going to reclaim, unless we're really overabundant in all of these things, we're going to reclaim our amino acids, 100% of those. We need those. Those are nutrients. We'll reclaim vitamins and minerals. We're going to bring back everything that we want in the body. We could reclaim some bicarbonate ion if we need it. We'll reclaim some urea if we need it. So again, the kidneys are adjusting your blood and urine concentrations of various things dependent on need. So if we are needing these things, then we'll bring them back. If we're in excess, then we'll excrete them. So, you can take 5,000 milligrams of vitamin C. So in that case, we're not going to be reabsorbing a ton of vitamin C, we'll be excreting it. But, so just kind of think about that when this is all going to be according to need. But what we're doing here in the proximal convoluted tubule is bringing like 65% of it back. There's a lot in there that we need. So that's a big important job. So the proximal convoluted tubule is making most of the adjustments to urine. It's making like 80% of the adjustments to the fluid that's passing through so that your blood can have everything that it needs and so that we can be excreting everything that we need to in the urine. So it's going to be reabsorbing like 65% of the materials. All of your nutrients, glucose, vitamins, minerals according to need, amino acids, and then it will be bringing back things like urea as needed and bicarbonate ion. So remember, the kidneys act as a buffer system, so kind of all of those adjustments, or most of those adjustments, are being made right here in the proximal convoluted tubule. Okay, so let me ask you a question. The cells in the proximal convoluted tubule do a lot of active transport. To serve this function, you would likely find increased blank in these cells. A, mitochondria. B, centrioles. C, lysosomes. D, all of the above. All right, yeah, the answer is A, right? Because mitochondria make ATP. Active transport requires ATP. That's what makes it active transport. So these cells have more mitochondria because the specialization is that they need a lot of ATP. So they can get those active transporters going and pump things against their concentration gradient. Okay, so what does our tubular reabsorption look like in the nephron loop? So the nephron loop has the important job of setting up the osmotic pressure gradient that's going to allow for the most amount of water reabsorption in the collecting duct, which is not part of the nephron. But just be aware of that. So nephron loop sets up the osmotic pressure gradient for water reabsorption in collecting duct. And it's got two parts, the descending limb and the ascending limb. The descending limb is permeable to water, but not salt. So we will do reabsorption of water in the descending limb. And then the ascending limb is permeable to salt, but not water. So we'll do reabsorption, active reabsorption of salt in the ascending limb. So as far as our tubular reabsorption goes in the nephron loop, we'll say in the descending limb, we're reabsorbing water. And this water is being picked up in the vasa recta. So we'll just put that in our pocket for now. And then in the ascending limb, we'll have active reabsorption of salts, and those are going to be reabsorbed to the interstitial fluid. So we'll reabsorb our salts to the extracellular fluid. And so primarily sodium chloride, but we can also bring potassium, pump potassium out to the interstitial fluid there as well, which would be reabsorption. And then when we get to the distal convoluted tubule, we'll just say that our reabsorption is under hormonal control. Okay, so for tubular reabsorption, 65% of the material that was pushed out in filtrate is going to be reabsorbed in the proximal convoluted tubule. When we get to the descending limb, we'll reabsorb some water that's going to get picked up in the vasa recta. We'll reabsorb, and when we get to the ascending limb, we'll reabsorb some salt that's going to get reabsorbed to the extracellular fluid. That's going to create this nice osmotic pressure gradient for water reabsorption. And then when we get to the distal convoluted tubule, all of our reabsorption will be under hormonal control. So let's think about it. What are things that we've said the kidneys can reabsorb in response to some hormone? Well, if the blood is low in calcium, we can reabsorb calcium. What hormone stimulates that? Parathyroid hormone. Okay, if we're low on water, we could stimulate water reabsorption. What hormone stimulates that? The antidiuretic hormone. Good job. Okay, if we're low on sodium, we could have a hormone stimulate sodium reabsorption. What hormone? Aldosterone, and water is going to follow that. Okay, so at the distal convoluted tubule for reabsorption, we could say that parathyroid hormone is going to stimulate calcium reabsorption. We could say that antidiuretic hormone is going to stimulate water reabsorption. And aldosterone is going to stimulate sodium reabsorption. Chloride ion is going to follow. Water will also follow. But directly, that's what's being stimulated, the reabsorption at the distal convoluted tubule. Okay. What about tubular secretion? So again, the proximal convoluted tubule is making most of the adjustments to our urine. So most of our secretion is going to occur here as well. Secretion. What's secretion? We said that tubular secretion is going to lead to excretion. So this is going to be, now if we look, where we're pulling things out of the blood and bringing it and concentrating it into this our tubular fluid that's becoming urine. So I think I keep changing colors. My most sincere apologies. I'm confident that you can work it out. So tubular secretion is the process by which now I'm going to pull things out of the blood and concentrate it in this fluid. So this is how I'm going to rid the body of unwanted wastes that are floating around in my blood. So again, the proximal convoluted tubule is making most of our adjustments to blood and urine. So most of our tubular secretion is going to happen here. So what am I going to pull out of the blood? Wastes like ammonia, and this is also where I'll pull out like my excess, if I've got excess hormones or excess vitamins or excess minerals. So drugs I can pull out here. I can pull out creatinine, like, you know, just all of the waste that are floating around in my blood. I'm going to pull out of the blood and concentrate them in the fluid that's passing through here. So for tubular secretion in the proximal convoluted tubule, we'll say this is going to do most of the tubular secretion. Things that we're going to secrete here, this is going to be our wastes. What are our wastes? Like ammonia. I think that's NH3+. Depending on what's going on with need, we could secrete urea here. We could reabsorb urea here also. There's this whole urea recycling thing that we're just not going to have time to deal with right now. So other things that we would pull out. Some drugs, anything in excess. That's really going to be the goal, is to pull it out of the blood and concentrate it. Now when we enter the nephron loop, nothing is going to be secreted. Except for, well, not in the descending limb. And then when we turn around and come up the ascending limb, there's a little bit of urea secretion. So that's part of urea recycling, again, that we're not really going to go through. But you can just be aware that urea can be secreted into the ascending limb of the nephron loop. And then in the distal convoluted tubule, again, we're under hormonal control. So anytime we've said, oh, if there's an excess of this, so you can stimulate excretion of the kidney. Well, if there's an excess in calcium, what hormone is going to stimulate calcium excretion? Calcitonin. And then potassium moves the opposite direction of sodium. So if we're reabsorbing sodium, then we're going to secrete potassium. So what hormone would stimulate potassium secretion? Aldosterone. So at the distal convoluted tubule for secretion, we could say calcitonin is going to stimulate the secretion of calcium, and aldosterone will stimulate the secretion of potassium. And this ability to secrete potassium under the influence of aldosterone is so important that that's actually one of the ways the zona glomerulosa can respond. If you recall, when we talked about the endocrine system, we said that the zona glomerulosa could release aldosterone in response to angiotensin II or in response humorally to an increase in potassium. And this is why. Because if there's an increase in potassium, then you can't repolarize cells, and cardiac arrest is not long off. So it's better to be able to secrete potassium than have high levels of potassium. And so if you're hyperkalemic, then you would secrete aldosterone so that you can stimulate potassium secretion. So then we would pull this potassium out of the blood and concentrate it in this urine. Okay. Also here in the distal convoluted tubule, if need be, this would be an area where we could take out hydrogen ions if our blood was acidic. The proximal convoluted tubule could do that as well. If our blood was acidic, then we could take out some hydrogen ions. Also, if we were basic, we could pull out bicarbonate ions. So really, the tubular secretion is going to be dependent on what's in the blood. But if it's a waste or we've got too much, then we're going to secrete it. And most of that secretion is going to be done here in the proximal convoluted tubule, because here in the nephron loop, we're hardly secreting anything. And then in the distal convoluted tubule, that's going to be under hormonal control. Really meaning that the proximal convoluted tubule is really doing most of the adjustments to this fluid so that we can become urine. Okay, so the regulation of our urine concentration and volume is determined by the body's need of water, but what's going on here is that we're reclaiming all of that water in the collecting duct that's over here, and the collecting duct is passing through this salty interstitial fluid. And so if you can imagine, we've got this collecting duct running here, and it's going to drain these collecting tubules, which are draining our distal convoluted tubules. All of our collecting ducts are going to drain down into papillary ducts, and when I get here in the papillary duct, I am urine. So this is urine, and there's nothing else that can be done to it. The whole way down this collecting duct, I am passing through this salty interstitial fluid, and I'm going to be reclaiming water. So how I am setting up this concentration gradient is here in these nephron loops, the juxtamedullary nephron loops in particular, that are dipping down into the medulla, and there are a couple things going on. If we look, we set up that what are two kind of systems that are going to help to increase the concentration of the interstitial fluid here so that we can have maximal reabsorption of the water here. So the countercurrent multiplier and the countercurrent exchanger. Counter, opposite, current, flow, multiplier. We're multiplying the effect. Okay, so the multiplication effect happens as we multiply the effect of actively pumping out salts up here. So the descending limb of the nephron loop is going to be reabsorbing water. And that water is going to be picked up in these vasa recta, which is where this countercurrent exchanger comes in. And the blood in the vasa recta is flowing in the opposite direction, which is where the countercurrent comes from. And then the exchange is allowing for this exchange of water, which is allowing for this multiplication of the effect of pumping out salts here. So what happens in the descending limb is that we reabsorb water to the vasa recta. And then when we get to the ascending limb, we reabsorb salt to the extracellular fluid. So what that's going to do is get it really salty out here. So if we think about it, the fluid coming in here is going to have, it's going to have its water and its salt. And as we pump the water out here, it's going to get saltier. So as we pump the water out here and pick it up in the vasa recta, we get saltier and saltier as we get down here. So we're super salty when we get down here. And then when we come back up here and we start pumping out the salt, now this fluid moving through here is dilute again, because the salt is all out here, getting trapped in this interstitial fluid. It's not being picked up in the vasa recta. So it's multiplying this effect as we go up this loop so that we get saltier and saltier. So that if you look at the tip of the loop, this is actually where we're the saltiest. And so what happens then is we actively pump out those salts, this fluid gets re-diluted as we come up in here. Then when we get to the collecting duct, this fluid is dilute relative to here. So now as I pass back through this salty interstitium, I can have maximal water reabsorption because I've created that osmotic pressure gradient. So I hope that makes sense. There are definitely other videos that you can watch that show it, like draw it out in an illustration and have it moving and talking to you all at once. I can only draw this picture and tell you. I can't illustrate it and move it all at the same time. So maybe watch one of those. But again, the countercurrent multiplier is established as we have this fluid moving through the nephron loop in opposite directions. The countercurrent exchanger happens because the blood flowing through in the vasa recta is flowing opposite of that, and it's exchanging this, we're having this exchange of salt and water, so that we're picking up the water here in our blood vessels, and we're concentrating the salts out here, leaving it in the interstitial fluid, and that allows this to be really salty. So here's your brain break. Kangaroo rats who live in the desert have to be able to reabsorb as much water as possible from what would become their urine, because they're dehydrated. They run around in the desert. And so our blood is about the same osmolarity as salt water. So if we drink salt water, it actually dehydrates us. But kangaroo rats are actually able to get water out of salt water. They're not it's not a salt kind of thing that's happening, and it's because they have these really huge juxtamedullary nephron loops that dip so far down in the medulla that they are able to just set up such an osmotic pressure gradient that they can actually get water out of salt water, out of seawater, which is pretty crazy. All right.
I did not know that, Mama.
So the formation of dilute or concentrated urine is going to be really dependent on the amount of antidiuretic hormone present in the system. So antidiuretic hormone creates these things called aquaporins in our distal convoluted tubule and collecting duct, and aquaporins are going to allow for water reabsorption. So as you're passing through this salty interstitium, how much water reabsorption you do, or how much we pull out of this fluid, is going to depend on how much antidiuretic hormone you have. So let's just imagine if we were so dehydrated that we had the maximal kind of secretion of antidiuretic hormone from the hypothalamus so that we could get the maximal water reabsorption here, then what kind of urine would we secrete down here? Would we have a lot, a little? Would it be concentrated, dilute? If we have the maximal amount, so if we're so dehydrated that we have a lot of antidiuretic hormone, that means that we're so low in water that we have a lot of ADH, then what happens is we would put all these aquaporins in my distal convoluted tubule, but also in my collecting duct, because this is what's passing back through that salty interstitium, and now I will pull out as much water as I can. And what I will release then, or excrete from the body, is a low volume of super concentrated urine. So if I have so little water that I've got so much antidiuretic hormone, what we would do is we would increase aquaporin number. Shh, I'm almost done. And so we would excrete low volumes of really concentrated urine. You all know that. You have been dehydrated before and you've gone to the bathroom and it stinks and it's really, really yellow and there's not a lot of it. It's because you're reclaiming as much water as you can. Okay, well, what about the opposite case? What if I am so overhydrated that I have no antidiuretic hormone? How much urine am I making, and is it concentrated or dilute? Well, if instead is that I've got so much water, I'm like drinking so much water that there's no antidiuretic hormone, then instead of bringing back all this water as I pass through the collecting duct, I'm just going to excrete it. And I would excrete a large volume of dilute urine. So excrete a large volume of dilute urine. So the loop of Henle, or the nephron loop, sets up the osmotic pressure gradient that allows for water reabsorption, but truly what determines how much water reabsorption you're going to do is how much antidiuretic hormone is present, because that's going to determine how much is coming out of that fluid now and back into the interstitial fluid. And then where is it going to go? Into the vasa recta and back to the body. So this will increase our blood volume. If we're bringing back this water, that would increase our blood volume, decrease our urine output. If I have no antidiuretic hormone, then I'm going to be pulling out all that water and peeing out a very dilute, large amounts of dilute urine. All right, good deal. We're almost done. All right, urine. So you can actually tell a lot about the health of a body from what you find in the urine. So this actually, like early on, one of the tests for diabetes was tasting the urine. And if it tasted sugar-y, and if it tasted sweet, then they knew you had diabetes because you were spilling over glucose. You weren't bringing it back into your body. Kind of interesting. This can also tell you about, like, if you've got proteins in it, then you know that there's something wrong. Probably a blood pressure problem because you shouldn't be pushing that out. So if we think about what's the chemical composition of urine, it's got water, but it's also high in waste. So it's got high amounts of waste. It's got urea.
My things are smelled like that.
It's got uric acid. It's got ammonia. And it's got everything else we're excreting from the system. So it should not have, it shouldn't have glucose. It shouldn't have proteins. It shouldn't have cells or anything like that. So actually that was one of the things that made this guy come to the world 3 weeks early. At the end of my gestation with him, 37-week appointment, that morning I woke up and I had a headache. And my head felt like it was full of cotton. My face was a little swollen. And I went into the doctor and the nurse took my blood pressure and she says, oh, it's 104 over 90, over 88. Great blood pressure. And I thought, well, you're not looking at my chart, because my blood pressure is like 90 over like 54. And so my blood pressure is low, so it was high for me, still in the normal range. She thought it was a great blood pressure. So the nurse leaves, the doctor comes in, and she's doing my exam, and I had all these questions that I needed to ask her, so I'm going through my list. And as she's measuring my fundus, I remember, what about, why is my face swollen? She said, well, swelling can happen when you're pregnant. Well, yeah, but what about this headache? She's like, well, that can happen too. So she finishes the exam and she goes and sits down and she says, tell me a little bit more about this headache. And I said, well, it feels like my head is full of like cotton. And she said, okay, I need you to do a 24-hour urine protein screen. So it was the worst test of my life. You have to collect every drop of urine that exits your body. And so it's just a nightmare. When you're pregnant, you're peeing all the time. So I got these 2 big gallon jugs of pee that I had to bring in the next day. And I bring them to the lab and go home. And I don't hear anything from my doctor. It's like 4:30, so I call the nurse. Hello, have you heard anything back? She says, no, so we'll just see you on Monday for your 38-week appointment. Okay, great. So I go about my business, and about 8 or 8:30 at night, my doctor calls and tells me that the lab has called her and I need to come in right away for her to take my blood pressure. Why? You're spilling proteins in your urine. Well, I'm not coming in. It was like the 22nd of December. He was due January 13th. I did not want to have a baby anywhere near Christmas. And so I just did not want to go in. I said, no, I'm not coming in. She said, yes, you have to come in. And I said, no, I'm not. She said, I need to check your blood pressure. And so I'm just going to check your blood pressure. And then you can go home. And I got in and I did not leave. And this guy was born on Christmas Eve. And my blood pressure it never reached preeclampsia levels. But for me, it was so high, I actually developed this life-threatening condition called HELLP syndrome. And he had to be delivered 3 weeks early. But the whole point is that you can look at what's in your urine and tell what's going on. So even though I had this normal blood pressure, it was too high for me and I was spilling proteins in my urine. And so my doctor could tell, and it probably saved our lives. So it's kind of cool. Physical characteristics, it's heavier than water. It's straw colored. That's all I have to say about it. All right, so after urine is produced, it is going to be conducted out of the kidneys into the ureters. So our ureters conduct urine to the urinary bladder. So they conduct urine to the urinary bladder. The bladder, if you recall, the bladder and the ureters actually together are the only location where you find transitional epithelial cells. So in the mucosal layer, we've got transitional epithelial cells. And remember, these are the cells that are going to transition in shape from cuboidal looking when the bladder is empty to more squamous looking as the bladder fills. And so they contain transitional epithelium on the inside, and then their intermediate layer is this layer called the detrusor muscle. And this is smooth muscle that you can forcefully contract to expel urine. Go figure that one out. So you can forcefully contract to expel urine. And then on the outer wall, on the top, we've got serosa, on the sides and bottom. On the lateral and inferior surfaces, we've got adventitia. What else do we want to say about it? If you look at it, the ureters come in to kind of the superior and posterior aspect of the bladder. And then the urinary bladder is going to lead down like this, and then it leads out through the urethra. So the urethra is the organ that's going to conduct urine out of the body. The storage capacity of the urinary bladder is about 800 mils. So it can hold about 800 milliliters. So the urethra is going to conduct urine to the external environment. And it's got 2 sphincters. Sphincters are little circular arrangements of smooth muscle that control the passage of material from one organ to the next. The internal urethral sphincter is all smooth muscle. And so it's involuntary. The external urethral sphincter has some skeletal muscle. So you can train this so that you can be potty trained. What you're doing is learning to control those skeletal muscle fibers in that external urethral sphincter so that you can hold it until it's convenient to void. So if we think about it, the ureters are bringing the urine into the urinary bladder, and then the urethra is conducting that away. So at the base of the bladder is the internal urethral sphincter. For females, there's a little bit of the urethra before we get to the external urethral sphincter. And so you've probably heard, that females lose bladder control over their life as they get older, and that's because you're putting so much pressure on your bladder over time that you kind of just wear out the ability to control that. In males, their internal urethral sphincter is right there at the base of their bladder, but then their urethra has to pass through their prostate gland before we get to the external urethral sphincter. So there's not as much pressure on that, so they don't tend to have as many kind of failures of the external urethral sphincter as females do. So just kind of interesting thing to be aware of. So micturition, that's the reflex that's going to control urination. And so micturition is urination, and it's a reflex. So when your bladder fills, it'll send sensory information back. So as your bladder is stretching, it'll send the sensory information back to your spinal cord, because spinal reflexes are handled not from the cerebrum, right? We handle them all through the spinal cord. So we'll send the sensory information back through the spinal cord. The spinal cord will come and relax our internal urethral sphincter. If you haven't trained the micturition reflex, if you haven't trained that external urethral sphincter, then you'll just go ahead and void. If you have trained it, then these signals, you're also going to be getting signals up to your cerebrum that you can feel this. So I can send down a voluntary command to control those skeletal muscles, and I won't void. And then I run to the bathroom, and now it's convenient, so I can relax that external urethral sphincter and I can go. So what do I want to know about micturition? It's a reflex. So an increase in stretch of the bladder is going to send sensory information to my spinal cord. In my spinal cord, I'll send out efferent information and relax the internal urethral sphincter. What's going to happen then is some urine is going to enter the urethra, and if you've got control, you can squeeze and hold that external urethral sphincter. So we'll relax the internal urethral sphincter. Urine will enter the urethra, and you either control or don't the passage of material through, or the passage of urine out of the external urethral sphincter. All right. That wraps up the urinary system. It's been fun. The hydrostatic pressure in the glomerular capillary is axillary.